Welcome back to our academic sessions. For the next talks, going to be the topics of gastric cancers, and we have many international leaders in stomach cancers for let you know about the important and clinical significance of these cancers in the world. Very first, I'd like to invite Professor Miftah and Professor Mégraud to share this session. As you may know, Professor Mégraud very well. He is a world leader in Helicobacter pylori infections and also with stomach cancers. And Professor Miftah is a vice president of the University of Atlanta in international affairs. And he also is a leader in Helicobacter and upper GI disease. Professor Mégraud and Professor Miftah, please.
Thank you very much. So, good morning, ladies and gentlemen, dear colleagues. So, it's my pleasure to introduce the first speaker, who indeed is Professor Varocha Mahachai. All of you know, she is from Bangkok and she is more or less organizing also this meeting. So, Professor Varocha Mahachai is a past president of the Gastroenterological Association Thailand. She is currently an advisor to the Bangkok GI and Liver Center, Bangkok Hospital, and a GI director at the Sukhumvit Hospital and Thonburi Burumuang Hospital. She is an academic advisor at Center for Excellence in Digestive Diseases, Thammasat University, and Science Research and Innovation. She now serves as committee members in various international societies, council member, chairman award committee, ASEAN Pacific Gastroenterological Association, and Dr. Mahachai serves on an editorial board of Helicobacter. In addition, she is a corresponding fellow in Asia for the Euro-American Helicobacter and Microbiota Study Group. She is also a member of the WGO Governing Council, chair on promotion of education in the Asia-Pacific region. Her major interests include all areas of the digestive system in GI, hepatology, endoscopy, with special interests on UGI disorders, Helicobacter pylori, GERD, esophageal cancer, colon cancer, IBS, constipation, IBD, and digestive health and microbiota research. So, Varocha, please.
Thank you for your kind introduction, Francis and Dr. Mehta. So, ladies and gentlemen, so I will start this session by presenting the global prevalence of Helicobacter pylori infection. As you can see, prevalence of Helicobacter pylori varies from different regions of the world. With the high Helicobacter infection rate, so mainly focused in certain parts of the world, like in South America and perhaps some countries in Africa, and maybe you can see high prevalence in a region like Bhutan, for example, and some parts of Eastern Europe. Like in Asia, it ranges from, can be low or moderate. Like in Thailand, for example, it's about 30 to 50% that we have Helicobacter pylori infection. Other part of Asia can be high as 50 to 70%. And you can see we have the data from ASEAN region. From ASEAN region, in Thailand, as you can see, around 30%, and perhaps high in Vietnam, up to maybe more than 70%. And this we compare with gastric cancer prevalence. Interestingly, in spite of high Helicobacter pylori over ASEAN countries, the gastric cancer rate were lower in most countries. You know, like 1.3 to 7.2 per 100,000 compared to 50 to 80 in China and Japan. Some countries, Vietnam, Myanmar, Brunei, Laos, tend to have a higher rate of gastric cancer, but it's still considered to be intermediate prevalence. This is, it's estimated that about 50% of the world population is infected by Helicobacter pylori, seem to be the most common human infection. And we look at the study in Thailand, as I say, 35% on the average have Helicobacter. It ranges from region to region, seem to be highest in the north and the northeastern part, but lower in the south. And I just like to present, because when you look Helicobacter, how you treat it depends on, as you heard before from previous speakers about antibiotic resistance. And this is our previous study report many years back, and we found the two antibiotics of interest will be metronidazole and kerathromycin. In 2013, it's only 3.7%. And if you look at the latest data in a couple of years, kerathromycin resistance has gone up to about 14%, but it's still below 15%. When you have a cutoff of 50%, how you select the regimen. And the others like amoxicillin and tetracycline have very low resistance, not more than 5%. And this is a prevalence of H. pylori in different ASEAN countries that report in the consensus back in 2016. In Thailand, average about maybe prevalence of Helicobacter around 30%, but it varies from different regions. Cambodia, 30 to 50%. Laos is also high, 68%. Myanmar, we surveyed before, is also high infection rate. Vietnam, also high. Others, countries in South Asia may be medium, 22%, 20%, Singapore, Philippines. So this is data in the ASEAN countries. And when you look at the antibiotic resistance pattern, the one of interest are kerathromycin and metronidazole. Metronidazole have been high in all countries, but as was alluded before, but it can perhaps become by use higher dose of metronidazole or use bismuth regimen to overcome metronidazole resistance. Kerathromycin, the cutoff is 50%. If you below 15%, you still can use a standard triple therapy. And Thailand is 15.8, but countries that high kerathromycin resistance include Vietnam, Indonesia, and perhaps Singapore. And like tetracycline and amoxicillin, I say, tend to be low in many countries, but interestingly, amoxicillin has high resistance rate of amoxicillin. These are data from Professor Mégraud that antimicrobial resistance in European countries and tend to have a high kerathromycin resistance around 26, as I say, if over 15 standard triple therapy or with regimen including kerathromycin should not be used. So whether you have quadruple or you have to modify the regimen, whether pre-treatment susceptibility test, whether it can be done, so that remains to be seen. Amoxicillin never a problem. You can repeat it, but except in amoxicillin allergy, level of fluoxetine is getting higher. Some use as a second line or third line. And also interestingly, Dr. Mégraud has data on lafabicin, which we haven't included in the regimen. And this is when we look at the Helicobacter pylori, we also interest in the gastric cancer prevalence. Helicobacter pylori tend to be high throughout ASEAN countries, as we report before. Although the cancer prevalence varies higher in Vietnam, Myanmar, Singapore, and lower in Cambodia, Philippines, and the lowest is in perhaps Thailand, Malaysia, Indonesia, less than 5 per 100,000. And so countries in Asia Pacific, in fact, I guess perhaps by the leader like Dr. Squano report before, we divide depend on the risk of gastric cancer. China, Japan, Korea have just standardized rate over 20 per 100,000, so it's a high risk. Although Helicobacter tend to be high in almost all these countries, but cancer rates seem to vary. Intermediate risk countries like Malaysia, Singapore, Taiwan, and Vietnam have more than 10 over 100,000, and low risk like Australia, India, and Japan. In spite of very high infection rate, especially in India, they have very low cancer rate. That's what we call enigma, gastric cancer enigma. We don't understand why, but it remains to be seen. But now, as Professor Sano talked about before, now from the Kyoto Consensus, Helicobacter pylori infection is an infectious disease, so it needs to be treated and eradicated despite of clinical situation. So I think that's all I would report to you today. Thank you for your kind attention.
Thank you. Oh, thank you very much, Professor Mahachai. Okay, so
we go to the next presenter. I'm happy to introduce my sensei because I graduated from his department, Professor Yoshio Yamaoka, MD, PhD from the Oita University. Recently, he is the Vice President and Trustee Oita University, Japan, and also a publication more than impact factor 2,400. Welcome to Professor Yoshio Yamaoka about H. pylori and the pathogenesis of gastric cancer, please.
Okay, thank you very much for inviting me to talk. Today I would like to discuss about the H. pylori and the pathogenesis of gastric cancer. And as you can see, this is an estimated age standard incidence and mortality rate in 2020. Now it's sorted by incidence. Mongolia is the worst, and Japan is the second place. However, if it's sorted by the mortality, you know, Japan is disappeared, and Mongolia is still the number one. If we check all 50 countries, Japan is appeared again. But as you can see, Japan is also very different from other countries. The incidence is very high. However, the mortality is very low. This is, of course, probably due to the advanced technology of the endoscopy, also the recent eradication of H. pylori. As you can see, the H. pylori is a gram-negative bacterium living only in the stomach and discovered in 1983 by Warren Marshall and class 1 carcinogen. So, for example, in Japan in 2013, the national insurance is covered to the all H. pylori positive gastritis patient. So before that, every year 600,000 people are eradicated. But after the insurance is covered, it's become 1.1 million people every year were eradicated. And finally, so around recently, incidence of gastric cancer is also decreased. So again, this time my topic is pathogenesis. So of course, it's various factors of H. pylori, also inflammation and host genetics, and environmental is also important. And recently, the gastric microbiome is also important. Okay, first I discuss about the virulence. So this is my old review paper in Nature Reviews. As you can see, there are, for example, the caregiver, and Dupay, these are well-known virulence factors. especially the well-known is a Kage. As you can see, the H. pylori cannot go into the cell, only attached to the cell surface. And using the type 4 circulation system, the Kage is going to the cell, and it is phosphorylated, and then several oncogene in the human oncogene is attached, and then induce a gastric cancer. And this might be the current consensus. And as you can see, in the clinical isolate, there is positive H. pylori and negative H. pylori. And within the H. pylori, positive H. pylori, there are also two types, East Asian type and Western type. This is very different on the 3' repeat region. The structure is different, especially the API D or C. So, and also there is some repeat in these regions. This binding activity is much higher in East Asian type compared to Western type. This is in vitro study, so that's why probably that's why the East Asian country gastric cancer is very high. So this is a consensus. However, some very interesting finding from our study. We have some collaboration with Mongolian doctors, and I got a short license and I went to 5 locations throughout the country. And very, very surprisingly, you know, it's a Western country, and the worst instance and mortality of gastric cancer. However, 80% are Western type Kage, and East Asian type is only 3.2%, and around 17% are KAG negative. However, in the KAG negative cases, all are gastritis. So probably the presence of KAG might be important. However, Western or East Asian, this is still doubt. So because all the cancer cases are Western type. Maybe the East Asian type KAG induce a severe inflammation, atrophy, and a high risk of gastric cancer. In Western countries, KAG negative, Western type KAG, and so the moderate inflammation and atrophy, so the risk of cancer is low. So in case of Mongolia, to tell the truth, it's really in the Mongolian stomach. Atrophy is not so severe. Also mild inflammation. However, high incidence of gastric cancer. So it means probably other H. pylori virulence factors or microbiomes or environmental factors is still very important. So first, check the environmental. This is in Mongolia. Even in Mongolia, in the Ubus, the country in the west side is a very high incidence of gastric cancer, even within Mongolia. And the H. pylori incidence is similar in all over the country. Everywhere very high, it's 80% infected. However, in the Ubus it's very high, and it's very parallel to the salt intake. So it's really, H. pylori is of course important, but salt is really important. It means environmental factor is also very important. And how about the microbiome? So this is, we also check the Mongolian populations. As a result, this is one of the very interesting findings. It's a blue color is a so-called Helicobacter, maybe H. pylori. So in the gastritis, atrophy, most of the predominant bacteria is H. pylori. However, in the cancer, gradual intestinal metaplasia, almost 50% of the intestinal metaplasia cases are already H. pylori is almost disappeared. And in gastric cancer, it's also H. pylori is, of course, some cancer cases, all H. pylori. However, in some cases, H. pylori disappeared, and black color Enterococcus and the Lactobacillus pink color is very predominant in some cancer cases. And so currently we pick up and isolate these bacteria, and finally we recently pick up the Enterococcus, so new Enterococcus bacterium from the stomach. So maybe in the near future will show the result. So whether this Enterococcus or Lactobacillus really induce gastric cancer or only because of the high pH and H. pylori disappeared, and then this kind of bacteria is appeared. And some of the interesting point is in the East Asian type Kage strain, it's only the N number is 5. However, the relative abundance of H. pylori is very high. So it means in the East Asian type Kage infected stomach, most of the bacteria is H. pylori. And so this is also somewhat interesting for us. And also recently we examined not only Mongolia and the Thailand, Bhutan, Cambodia, Bangladesh, Myanmar, Nepal, and Vietnam. So H. pylori positive cases and negative cases. So especially the negative cases is the rapid virus test negative, bacteria culture negative, histology negative, immunohistochemistry negative, and even serum antibody negative. So these complete negative cases, we check it. However, somewhat interestingly, the OTU is H. pylori negative cases in the blue color, but this is always in the discussion that some cases, even these patients have H. pylori using the 16S-ribosomal RNA. This should be not a contamination, so there is many similar paper from other group. And if the H. pylori positive cases, there are almost abundance is H. pylori is almost all bacteria is H. pylori. However, is by negative cases is many other bacteria. So the Chao index is significantly higher in negative cases compared to positive cases. And also that this is a distribution. It looks like unclear. However, for example, this is Nepal and Bhutan. Nepal and Bhutan is even the neighbored country. The composition of the bacteria is very different. Also, it's Cambodia and also Thailand. It's also the neighbored country. However, the composition is very different. So whether these differences are related to disease or not, so we are now checking. So from this summary, the current chronic H. pylori infection might be different. The local composition of the microbiome in the stomach and the H. pylori OTU were frequently found in the stomach in spite of their negative for H. pylori diagnosis test. So this is some transparent infection is occurred or not. We are now checking. And the microbiome differed among human population. However, it's still unknown about H. pylori negative gastritis. Further investigation is necessary. So in the gastric microbiome, still not so much clear. So some are difference. So what is really important? Then next we try to get a new H. pylori virulence factors. So for this purpose, we use so-called GBAS, genome-wide association study. So this is a recent publication. We performed the GBAS for investigating the genomic feature distinguish H. pylori isolate from the cancer and ulcer patient from East Asian type strain. So we only pick up the East Asian type strains because they are all Kage East Asian type positive. However, you know why? In some H. pylori positive patient become the antrum predominant inflammation and gastric duodenal ulcer. But in some patient, corpus predominant inflammation and gastric cancer. What is the difference? both are KRAS positive. Previously, we found the doppel, however, is not still there is controversial. So we checked the GBAS study. From the 600, we only pick up the actual, really the East Asian type, 240 strains, 125 gastric cancer, and 115 dural ulcer cases. And as a result, so we found totally the 11 genome changes, 8 SNPs, and also the 3 motifs. So this mutation is related to the distinguish between the cancer or dural ulcer. So for example, the first one, the DSBGK. So this is some kind of the related to the mortality. So currently, what is the difference? So because this is a main function is a mortality, so motility, and also the moving. So this we use some soft gel analysis. So we make the knockout. So if the knockout is completely the moving is disappeared, and then if we make the complemented strain, makes a complementation, its moving activity is recovered. However, if the complement to the gastric cancer gene or complement the dural ulcer genes, this is somewhat unclear. However, if we complement with gastric cancer this gene, the moving is more stronger than dural ulcers. So it means probably the cancer gene has a more activity to maybe go into the body. So that's why it becomes a gastric cancer or something. So now we are trying to investigate. So in conclusion, gastric cancer is an inflammatory inflammation associated malignancy, epidemiologically related infection with H. pylori, which is considered necessary but insufficient. And the combination of environmental and bacterial factor will increase the risk of gastric cancer. So environmental factor such as salt intake are also important. Currently, none of the pituitary prevalence factor alone is a disease specific. KRAS is important, however, maybe we need a combination. But recent technology for the next generation sequencing can be find the novel prevalence factors as well as prevalence microbiome in the stomachs. Thank you for your attention. So we currently, as you may know, so the maybe one more time the last slide. You can go back. It's okay. Okay. So this is our collaboration that I really went to there to do the endoscopy. So that's why, for example, the Miftah, he is my previously my PhD fellow in Indonesia. So we went together almost 20 locations in Indonesia. Also, of course, from Mahachai's Rick group, not only Thailand, but also the Myanmar, Bhutan, Cambodia, and Laos. So we went together, especially we have many strange and unforgettable experiences. Thank you very much.
Thank you, Professor Yamaoka. The question maybe in the last session.
Okay, so I'm pleased to introduce the next speaker, Dr. Jo Town Lin, which graduated from the School of Medicine, National Taiwan University. He received his MD, PhD at the Graduate Institute of Clinical Medicine in National Taiwan University in 1986. Currently, he is the Vice Superintendent, IDA Hospital. So he's also Professor Emeritus at National Taiwan University in Taipei. So maybe we can go to his talk concerning the risk stratification for gastric cancer before and after Helicobacter pylori eradication.
Thank you, Mr. Chairman. Dear colleagues, ladies and gentlemen, it's a great pleasure and honor for me to be invited here. This is the first time when after the COVID-19 pandemic, it's very cheerful for me to come to Bangkok. And the topic I will present is the risk stratification for gastric cancer before and after Helicobacter pylori eradication. In the following talk, I will talk about the relationship between gastric cancer and the H. pylori eradication. And also I will talk about the gastric cancer prevention program in Mahachai Island. Then we will go to the risk stratification of gastric cancer before and after H. pylori eradication. First of all, I will discuss about the cancer and H. pylori eradication. As shown in this slide, you may know that there are so many environmental factors which can induce gastric cancer. For example, the cigarette smoking, drinking, salted food, smoked food, even salt itself, and bacon, sausage, they all contribute to the development of gastric cancer. More recently, they talk about obesity or even overweight can induce gastric cancer. However, in recent concepts, Helicobacter pylori infection will be a very important, crucial environmental factor to induce gastric cancer. Therefore, there are so many ways to prevent gastric cancer. For example, intake of fruit, sausage, fruit, and vegetable, and even curry, garlic, green tea are reported to be helpful to prevent gastric cancer. Of course, some of the drugs, such as aspirin, metformin, or NSAID, are also chemopreventive agents. Today, we more focus on the eradication of H. pylori to prevent gastric cancer. So I will talk about a long story from 1995. At that time, we start the gastric cancer prevention program in Mahachai Island. Mahachai Island is a small island in between China and Taiwan. This is a community with high risk of gastric cancer. The annual incidence of gastric cancer is about 5 times 4-fold higher than that in Taiwan. The annual incidence and mortality rate of gastric cancer are highest in Taiwan at that time. Therefore, we have conducted 2 sequences of gastric cancer prevention program. The first one, the original one, started from 1995 to 1998. At that time, we used the serum peptinogen test as a serological test, and then we identify someone who are at high risk to develop gastric cancer. Then we do the endoscopic screening for this subject. Between 2004 to now, we change the strategy. We test and treat H. pylori, and then we do the endoscopic examination. So we talk about the story in between 1995 to 1998. During this period, we start to include the subject who has positive result of serological test for pepsinogen. And also we check the Helicobacter antibody, and then we do the endoscopic examination. This is a very old photograph. We check the blood by using the serological test for pepsinogen 1, pepsinogen 2. And we do the fiber scope, not the video scope, for the volunteer or the patient with high risk after the examination of serological test for pepsinogen. And this is myself in the right lower column. It's very young at that time. You can see that we use the gastro fiber scope examination. And then this kind of screening test has been performed for 4 years, from 1995 to 1998. Totally, we have 3,541 subjects has been included after serological test for pepsinogen. 946 positive case has been asked to do the endoscopic examination. Only 2/3 of the subject, 663 subjects, received endoscopy. Among them, 2 gastric cancer has been found. The result is very poor because that the year rate of gastric cancer just by screening is very low. At that time, we have made a conclusion that pepsinogen test, that means pepsinogen 1 less than 30, pepsinogen 1:2 ratio less than 3. That means they might have the intestinal metaplasia or gastric atrophy, which will be at high risk to develop gastric cancer. We can use this kind of serological test to identify 946 high-risk subjects, and then we do the endoscopic examination. However, the year rate is very low. Only 2 gastric cancer was detected in 663 screening endoscopy, while there are 28 gastric cancer were missed. The result was very unsatisfactory. The endoscopy approach was constrained by population participation, the referred to endoscopic rate, the manpower of endoscopists, and the endoscopy imaging technology. What we used is the fiber scope. Then an alternate approach is mandatory required. So we talk another story from 2004 until now. The time period from year 2000 to 2004 to 2008, we do the test and the treatment of H. pylori infection, and then we do the endoscopic examination for 6 sessions. This is the story in 2004 to 2008. The left side means that we use the carbon-13 UBT test for screening for Helicobacter positive patient. And then we do the endoscopic examination in right upper column. You can see that the endoscopists usually use the video endoscope to detect any lesion in the stomach. And then we give the positive patient with the triple therapy, the classical triple therapy with omeprazole plus amoxicillin plus clarithromycin for 1 week only. However, the result is very satisfactory. The long-term change in gastric cancer incidence are shown here. By year 2016, we have observed a 53% reduction of gastric cancer incidence was found. And then we anticipate at year 2025, there will be a 60% reduction of gastric cancer incidence in the future. Therefore, there is no more any gastric cancer in Mahachai Island, we thought. And this is the meta-analysis of the result of Helicobacter eradication and the gastric cancer incidence. The overall, a 64% reduction of gastric cancer incidence rate was observed in all studies. Moreover, the benefit of eradication is greater for high-risk population, such as Japan, Korea, China, but not in a low-risk country. In 2019, on September 29 to 30, we have invited the expertise in the world, including all the guests and Professor Mahachai also. And we have invited them come to Taiwan, Taipei. We have the Taipei Forum. We focus on the Taipei consensus of H. pylori eradication and the gastric cancer prevention or gastric cancer prevention program. The final result of Taipei consensus has been published in GUT 2002. You can see that in the left upper column, you can see a building, the tall building, the Taipei 101 on the front page of GUT magazine. However, they still have some gastric cancer occur even after H. pylori eradication in Mahachai Island. You can see that the majority of the cancer were advanced gastric cancer. They may be too old, they may be too young, they are the new immigrant coming from mainland China, or they are EGD refuser. Only one patient had the early gastric cancer, which was detected by screening. So the reason why do gastric cancer occur after H. pylori eradication? The first explanation is gastric cancer may be caused by the risk factor other than H. pylori. For example, the EB virus or some genetic factors or other environmental factors. As you know, the gastric carcinogenesis are very complicated. The three major factors, the number one is pathogen. Except for Helicobacter pylori infection, EB virus or other microbiota can contribute to the gastric cancer formation. Number two is the host factor. The genetic susceptibility such as HDGC gene or HMPCC gene, they will also contribute to gastric carcinogenesis. The third one is environmental factors. The majority are the lifestyle, such as smoking, high salt diet, low vegetable diet. All of these kind of environmental factors may contribute to carcinogenesis of the stomach. Therefore, the first reason might be gastric cancer may be caused by the risk factor other than H. pylori. The second reason might be too late to eradicate H. pylori. If there is an irreversible genetic change of atrophic gastritis or intestinal metaplasia in the stomach, there is no point of no return. There will be too late to eradicate the H. pylori. This is the schema for the Korea concept of multi-step process of gastric cancer. When Helicobacter infection induced the normal stomach to the chronic active gastritis, and then they progress to atrophy, intestinal metaplasia, and then early gastric cancer and advanced gastric cancer. If we can eradicate the H. pylori, then we can stop the process in the chronic active gastritis. Therefore, we might have the conclusion that if there is no Helicobacter infection, there will be no gastric cancer. However, the story may not be true because that if there is an irreversible genetic change in the stomach when there is atrophic gastritis or intestinal metaplasia. At that time, if you eradicate the H. pylori, they still progress into cancer, and you cannot guarantee that no Helicobacter will no gastric cancer. Even no Helicobacter infection, they still develop gastric cancer. Therefore, the two reasons why gastric cancer still occur after H. pylori eradication. Number one is other factor. Number two is too late. So we want to have a new strategy to stratify the risk of gastric cancer after H. pylori eradication. We have done a two-stage screening program between 2015 to 2018. Because that the new case of gastric cancer still occur even after master eradication program, a two-stage screening program was implemented in 2015 to 2018 in Mahachai Island. The first stage is we use the serological test, the gastric panel. The gastric panel consists of Helicobacter pylori IgG antibody, gastrin 17, the so-called G17, the pepsinogen 1, pepsinogen 2. And then the second stage is those defined with the high risk will be referred to endoscopic examination, and we do the histological assessment of the gastric mucosa to evaluate the DNA methylation label, the microRNA 124A-3. As you know, if we can successfully eradicate H. pylori, the gastric mucosa will be improved. After improvement of the gastric mucosa, the cutoff value of pepsinogen test will vary a lot. You can see in this picture, the pepsinogen 1, pepsinogen 2 will significantly decline. However, the pepsinogen 1 to 2 ratio will increase because the gastric mucosa became more improved. Therefore, originally, we used the pepsinogen 1 level and the pepsinogen 1 to 2 ratio 30 and the ratio 3 is a cutoff value of the diagnosis for extensive pre-malignant gastric lesion or gastric cancer. The area under the curve will be only 68%. The accuracy for diagnosis the gastric cancer risk is very poor. Therefore, we have modified the new cutoff value for pepsinogen 1 as 45, and the ratio of pepsinogen 1 to 2 is 6. And then we can increase the area under the curve for the new pepsinogen 1 and pepsinogen 1 to 2 ratio. The area under the curve is 86%. The accuracy is much, much better. Another important investigation is we add the microRNA, the methylation marker of the gastric mucosa, as an important factor to detect the risk of gastric cancer. This is the study from Ushijima, and we have the help with this kind of methylation marker. Then we check using the methylation test for the risk of extensive pre-malignant and gastric lesion. And then we can have an area under the curve, 68%. The accuracy for gastric cancer risk will be increased much. So we combine the pepsinogen test plus the methylation test. Then we have the area under the curve, 90%. It's a good accuracy for gastric cancer risk and also very good for exclude the risk. So our conclusion is After H. pylori eradication, the cutoff value of pepsinogen test varied. The serological test alone can only predict the extent of atrophic gastritis and intestinal metaplasia. The risk of irreversible genetic damage in the gastric mucosa can be predicted by aiding the measurement of DNA methylation level, and serological test plus the methylation test may better discriminate between those at high risk or low risk to develop gastric cancer. Dear colleagues, we summarize our speech here. Before H. pylori eradication, we used the pepsinogen test plus the screening endoscopy. The year rate of gastric cancer by endoscopy was very low. It's not cost-effective. But after eradication, we used the pepsinogen test. We have modified the pepsinogen 1 and the ratio of pepsinogen 1 to 2. The ratio has been modified, plus the endoscopic examination for the gastric mucosa, and plus the DNA methylation label in gastric mucosa. They will have the good accuracy of gastric cancer risk prediction. It also can help us to detect the H. pylori negative gastric cancer. It is cost-effective. Thank you very much for your kind attention. Thank you very much.
Okay, question will be after, so we go to the next.
The next speaker is Professor Jae Gyu Kim. He is a professor of internal medicine at Chung-Ang University Hospital, and recently is a president of the Korean Society of Gastroenterology. The time is yours.
Thank you very much for a kind introduction, Chairman, and especially organizing committee. I have no disclosure. These are table of contents of my presentation. This slide shows the overall treatment algorithm of gastric cancer in Korea. The potentially resectable tumor, we do surgery, and the metastatic cancer or locally unresectable tumor do the systemic chemotherapy. In Korea, the endoscopic treatment is very popular. In proper indication, we do endoscopic treatment. My talk is based on the Korean and Japanese guideline for endoscopic resection of early gastric cancer and comprehensive treatment of gastric cancer. Endoscopic treatment is steadily increasing in Korea. In 2019, among the total of gastric cancer, endoscopic resection is 40.3%. In early stage, especially in early stage, the endoscopic resection is obviously the surgery. Endoscopic resection should be considered for tumor harboring a very low possibility of lymph node metastasis and suitable for en bloc resection. Endoscopic mucosal dissection is higher en bloc resection rate compared with the endoscopic mucosal resection. So we should be considered primarily ESD in endoscopic treatment. In Japanese guideline, here are the indications. Mucosal differentiated tumor less than 2 centimeters without ulcer in any size. And another thing is mucosal differentiated type less than 3 centimeters with the ulcer. And another thing is undifferentiated type cancer, mucosal cancer less than 2 centimeters without ulcer. This is absolute indication in Japanese guideline. In Japanese guideline, focus on the recently undifferentiated type integrated into absolute indication for ESD. based on the result of multi-center prospective study. Compared with the Japanese guideline, in Korean guideline, we do the absolute indication in endoscopic treatment, but in the expanded indication is almost overall the same with the Japan, but relatively lower recommendation and evidence level in expanded indication. A little bit conservative attitude in endoscopic treatment in Korea. This is the assessment of curability in Korean guideline. The absolute indication is en bloc resection, mucosal differentiated tumor without margin involvement, lymphovascular invasion, and ulcer. The expanded indication is almost the same of ESD indication, but another additive thing is differentiated types of mucosal involvement less than 500 micrometer, less than 3 centimeter included. Otherwise, non-curative resection, we recommend the surgery, but if a horizontal resection margin is positive, we do endoscopic management preferred. Endoscopic resection is based on the local factors and the risk factor of lymph node metastasis. In Japanese data, this is the absolute indication in green zone. The yellow zone is relative indication. Lymph node metastasis is negligible percentage in the endoscopic indication. Recently, Japan developed a cura system for the strategy of gastric endoscopic treatment. They do the curative resection and the non-curative resection. They applied now, but in Korea, still not yet. Move on to surgery and chemotherapy. In Korean guideline, usually in early stage, we do the surgery, D1 lymph node dissection and minimal invasive surgery, laparoscopic surgery. In higher than stage, D2 lymph node dissection and do open surgery. There are some discrepancies in neoadjuvant chemotherapy in Western and Eastern countries. In Korea, Japan guideline, the resectable cancer we do surgery. Especially neoadjuvant chemotherapy in the bulky lymph node case, in Japan. But in ESMO guideline, they recommend the neoadjuvant chemotherapy. Maybe report of positive efficacy of neoadjuvant chemotherapy, mainly in Western studies, due to larger proportion of G-junction cancer than Asian studies. In Korea, recent multi-center study was done. A better progression in free survival in neoadjuvant cancer group, but no change in overall survival. This slide shows the first-line treatment for advanced gastric cancer. In Korean guideline and Japanese guideline and European guideline is overall the same, but in HER2 is the target in HER2 positivity for trastuzumab, and in ESMO guideline, they check the recently recommended PD-L1, and if the positive, they do the immunotherapy. For the second and third line treatment of advanced cancer, the various chemotherapeutic regimen was shown like this. And after that, adding the target therapy and the immunotherapy, it can be done depending on the guideline. For the targeted therapy, molecular subtype are associated with distinct molecular and clinical characteristics. Clinical relevance of molecular classification has a potential for translation into precision oncology strategies. Novel agents, including receptor tyrosine kinase, monoclonal antibodies, and antibody-drug conjugators, are currently being tested on ongoing biomarker-guided trials. However, molecular heterogeneity of gastric cancer is a major reason for the frequent failure of biomarker-based clinical trials. This figure shows the TCGA classification and summarize molecular characteristics and the clinical characteristics. Maybe we do the understanding of these the characteristics, we move forward targeted therapy. This is the selected trials of biomarker targeted therapies. Now the HER2 positivity and MSI status and the PD-L1 expression is only established biomarker with the efficacy of therapies. HER2 positivity for trastuzumab and the MSI status for pembrolizumab and the PD-L1 expression in nivolumab. Now it's being used treatment and under research. Move on to immunotherapy. Checkpoint proteins such as PD-L1 on tumor cells and PD-1 on T cells help keep immune response in check. The binding of PD-L1 to PD-1 keeps T cells from killing tumor cells in the body. Blocking the binding of PD-L1 to PD-1 with an immune checkpoint inhibitor allows the T cell to kill tumors. Here are the many kinds of published or ongoing immunotherapies according to HER2 positive or negative. These are the ongoing drugs, PD-L1 inhibitors. Now, first trend is the combined together chemotherapy and immune checkpoint inhibitor. Many kinds of drugs being studied. We can get the data or result in the 2023 or in the near future. This is the Checkmate 649 study for the nivolumab. They evaluated combined positive score for the PD-L1 expression. In high combined positive score, the positive data of overall survival. These are the clinical trials in the first-line anti-PD-1 studies in HER2 negative gastric cancer. Only the nivolumab and the sintilimab has a positive data. They all has higher combined positive data. Another trend is combined together the three kinds of drugs. Is chemotherapy, immune checkpoint inhibitor, and another things. And now try with the nivolumab or pembrolizumab or another things. This is the trial, ongoing trial for lenvatinib. Lenvatinib is an oral receptor tyrosine kinase inhibitor with the activity against the VEGFR 1, 2, 3. Is ongoing data. Another ongoing data is for the bemarituzumab, is the monoclonal antibody of FGL2B antibody. They may be adding the targeted therapy combined together with the chemotherapy and immunotherapy will have a new trend. I will show the chimeric antigen receptor therapy. Chimeric receptor, for making the chimeric receptor, we do the single chain variable fragment sequence. Is for the specific antigen of the tumor. In this chimeric antigen receptor, bind together tumor. Together with the tumor cancer antigen, and then activate co-stimulatory domain and activate the T cells and kill the tumors. For the CAR therapy, we extract the T cell and make the CAR cells in the lab and After that, the grow, we do the infusion T cells into the patient, and the CAR T cells maybe bind to cancer and kill them. These are the targets of CAR T cell in gastric cancer. As you know, the solid tumor, the CAR T cell therapy is a relatively low response because immunosuppressive tumor microenvironment, and the tumor has evasion of immune system. We do introduce the conditions for ideal cancer antigen in solid tumor. It has a uniform expression on the surface of cancer cells and playing a decisive role as an oncogenic driver in cancer cells and not expressed in normal cells. Here are the clinical trials of CAR T cells for the gastric cancer, but as I told you, the solid tumor is a relatively low response. We are expecting the good result of CAR therapy in gastric cancer. Finally, I introduce the possible role of microbiota in the treatment of gastric cancer. Bacteria was traditionally regarded as carcinogenic, including Helicobacter pylori. But now the evidence for anti-cancer property of bacteria has emerged. Bacteria exert anti-cancer effect via multiple mechanisms, colonizing tumor, releasing substances, suppressing nutrients required for tumor metabolism and proliferation, serving as a vehicle for anti-cancer drug delivery, forming biofilms, and enhancing host immunity. This slide shows the overview of bacteria-based cancer immunotherapy. First, bacterial targeting, direct targeting of tumor, and next, naive cell bacteria can activate the immune system. And we can make engineered bacteria and connecting to the immunotherapy and for the cancer treatment. Finally, bacterial component can activate the immune systems. This is a kind of the example. This study was to discover novel immune-related biomarkers in gastric cancer using fecal and blood samples. These bacteria were associated with tumor response to nivolumab. This slide shows the discovery of gastric cancer-specific microbiome to predict the response to immune checkpoint inhibitors. In summary, the treatment of gastric cancer, of unresectable gastric cancer, has tremendously evolution. Mainstay was the cytotoxic chemotherapies. Now the targeted therapies is being tried and applied. And another new treatment is the immunotherapy is now applied and under research. New strategy is with the CAR T cell therapy and with the microbiota and another new therapies. Maybe precision medicine is keyword of new treatment in the near future. I would like to thank Professor Hwang and Park for helping me. Thank you for your attention.
So thank you very much. So we have been informed that because we are so late that the panel discussion is suppressed. So very sorry about that, but the time is now for the next session, I think.
We have several questions, especially in online also, because the time is. Thank you very much for this session.